All right, so Screenomatic only gives me 15 minutes, so let's not waste any time. Let's go full screen. Boom. Chapter 10 is about DNA, RNA, and protein synthesis, the structures and functions of DNA and RNA, and the building of a protein. This do now I assigned last week. You should have. If not, go ahead and do the Google form. Here it is right here. Click that link. There you go. You should have already watched the videos and read the article and answer the questions with it. Now, here are the people to know from 10-1. People and words. Frederick Griffith's experiment showed that hereditary material can pass from one bacteria cell to another. Oswald Avery showed that DNA is a hereditary material transferring information between the bacterial cells. Alfred Hershey and Martha Chase confirmed that DNA, in fact, and not protein, is hereditary material. Now, why is this important? Because this was, I think Frederick Griffith was like the late 1800s where his experiments were taking place. Oswald Avery was not too far after, but these two, I think after like 30, 40 years, decided to confirm what these two had already figured out, which wasn't commonplace information. We now know that to be true. But before we knew it to be true, somebody had the question about it. So these are the people to know. Virulent is definitely a term you might want to get familiar with. Think about the first four letters there, viru, close to virus, which is why we're in this situation. This disease-causing strain of bacteria. Transformation is not what you think, right? It's not like Transformation Tuesday. This is how genetic information is actually transferred from cell to cell or organism to organism. And imagine being a bacteria and then being infected by a virus. That is what you would call a bacteriophage. So, these were the three videos I assigned you to watch and kind of get an idea of how virulent strains and genetic information is being transferred, right? This is the questions that these guys were asking. So I'll pause for dramatic effect where you guys can pause the recording, watch the videos again, and now you should have a better idea of the article that we had earlier, okay? So, I'll give you there a couple seconds. Chapter 10, Section 2 was about the DNA itself and the structure of DNA. So I will turn these three do now questions into a Google form. All right. So okay, here's why I assigned a video and questions, because the DNA double helix, discovering that was one of the largest discoveries in human history, only because it took about four people, well, more than four, but took about four people who three of them got the most credit. So you'll watch the video and you'll understand why James Watson, Francis Crick, Rosalind Franklin, and Maurice Wilkins are important. More importantly, just a little bit of history about this woman here. This is Rosalind Franklin, who assisted these other three gentlemen and got the least amount of praise at the time. Obviously, it's important that you know that there are women and there are people of color women and women of color and people of color who contribute to science all the time. So it wasn't just, older white men, right? Now, parts of the nucleotide. More importantly, the parts of there are four DNA nucleotides. There's adenine, thymine, cytosine, guanine. Now, if you've been paying attention here, the parts of a nucleotide, there's a phosphate group, right? Which you'd normally see a P in the circle because that's for phosphate. There is a 5-carbon, 1, 2, 3, 4, 5-carbon sugar, also known as pentose, right? Pento means pento, right? 5, and os is sugar. So every nucleotide is made up of the phosphate group, the 5-carbon sugar, and a nitrogenous base. But the nitrogenous base is where things get different. You either are made of the phosphate group, sugar, 5-carbon sugar, and an adenine, or a cytosine, or a guanine, or a thymine. And if you happen to be an RNA nucleotide, you could potentially be a uracil nitrogenous base. Now I want you to look at the structure of a DNA. It's a double helix, right? Made up of a phosphate backbone. So the backbone itself is what allows us to have this double helix staircase looking thing. And the cool part in the middle is, look at this color coordination right here. Thymine, being purple, is going to be next to what? In teal, adenine. Guanine in blue is going to be next to what? In red, cytosine. So if you notice, they pair up. Now, DNA is double-stranded. RNA, single. You also have these things called purines and pyrimidines. Long story short, purines have 2 carbon rings. Pyrimidines have 1 carbon ring. That simple. Now look at what ones fall under pyrimidines and what nucleotides fall under, or nitrogenous bases fall under purines. Okay? So let's talk about covalent bonds and hydrogen bonds. First, we talked about covalent bonds and hydrogen bonds way back in probably like chapter 3, chapter 2. Now they're back. So when we talk about DNA, each of these nucleotides, which are only different because of these nitrogenous bases, each of these nucleotides on either strand, okay? This is one strand, and this is one strand, okay? Each nucleotide on each strand is held together by a covalent bond between the sugar on one nucleotide and the phosphate group on the next. So look, here is the adenine nucleotide. They said that it is held together by the covalent bond between the sugar, that's what this big S stands for, and the phosphate on the next nucleotide. Look at that. There's a phosphate. Here's another big S and another nitrogenous base. These two are held together by this bond right here. Now the bases, these base pairs in the middle, are in the center. They face each other, right? Because this is one strand and this is another strand. Now the bases of a nucleotide face the center of the DNA molecule, and the bases can form somewhere between 2 or 3 hydrogen bonds between the 2 strands. What do you think these dotted lines are? These are hydrogen bonds. Now, this is not a hydrogen bond. This is a covalent bond, solid line. So what did that tell you about the hydrogen bonds? They're easily broken, easily formed. Complementary base pairs. This comes from Erwin Chargaff, which is the guy who was also mentioned in the video I assigned. There are rules to the game. Base pairing is complementary. Complementary with an E means that they fit together like puzzle pieces. Complementary with an I is like if I said something nice about you. What happens on one strand, the exact opposite happens on the other. So if I have T, A, C, G on this strand, on the exact opposite, G always goes with C, G always goes with C, A always goes with T, T always goes with A. In DNA, adenine goes with thymine, adenine, thymine. Guanine always goes with cytosine, guanine, cytosine. In RNA, RNA is single-stranded, but adenine is going to pair with uracil. So look at RNA. Here's adenine, which should pair with thymine because this is a DNA strand. But this is an RNA strand, so you'd find instead of a T, a U, and a U should always go with A. Now this helps hold the bond between the RNA strand and the DNA strand, or the bonds between a DNA strand and the second DNA strand, the leading or the lagging. This also tells how DNA replicates during cell division, where one strand is going to serve as the template for making a new complementary strand. As a matter of fact, this is typically what happens. You have the DNA template and the RNA copy. I suggest you do this activity, right? This is where you get to build a DNA molecule using the pairing principle. This is your exit ticket for 10-2, right? So, I'll let you guys get to that one. Now, what I would have done in class is made you guys, I would have brought you pipe cleaners and we would have built our own DNA molecules. So you can definitely watch this. If you have materials at home and you want to do it yourself, go ahead. If not, if you don't have pipe cleaners, there's plenty of ways to do it. And I actually might make that still a project. So chapter 10, section 3 is about DNA replication. I only get about 15 minutes of per recording. So I'm going to probably break this down here, and we'll start chapter 10 and 10 section 3 and 10 section 4 in the next recording.